Hello friends, today we will learn about each artificial chromosome in transgenesis. It is the second video of transgenic animal. So let's begin. So in the conventional transgenesis, small sized transgenes that is 20 less than that is more than or equal to 20 kilobase pair are transferred through vectors. This involves a risk of losing some important sequences including the regulatory ones. Further, several genes exist as complexes and are too large to be handled by conventional vectors. Each artificial chromosome insert YACS can carry large size genes. That size ranges from 100 kilobase pair to 1000 kilobase pair and are effectively used in transgenesis. By using microinjection technique or transfection of embryogenic stem cells with YACS, transgenic mice have been produced. Now let's know about the examples. There are two examples. First is YAC are used to carry human beta globin gene complex, that is 5 gene with 250 kilobase pair size and produce transgenic mice. Second is YACs are employed for producing human antibodies in transgenic mice. Synthesis of antibodies is very complex process. YACs carrying varying sizes of DNA sequence, that is 800 to 1000 kilobase pair each artificial chromosomes have been employed to generate different antibodies. Now let's know about transgenic mice and their applications. Mouse, although not close to humans in its biology, has been and continues to be the most exploited animal model in the transgenesis experiment. The common feature between man and mouse is that both are mammals. Transgenic mice are extensively used as animal models for understanding human diseases and for production of therapeutic agents. Adequate care, however, must be exercised before extrapolating data of transgenic mice to humans. Mouse models for several human diseases like cancers, muscular dystrophy, arthritis, Alzheimer's disease, hypertension, allergy, coronary heart disease, endocrine disease, that is a next is neurodegenerative disorders, etc. have been developed. Now let's know about the human mouse. The transgenic mice with human immune system were produced and they are commonly referred to as human mice. For this purpose, mice with severe combined immunodeficiency, that is SCID, a condition characterized by total lack of immune system cells, were chosen. Human thymus tissue from an aborted fetus was transplanted under the capsule membrane of the kidney of the mouse. Human lymph node was placed under the opposite kidney. After about a week, immature immune cells, that is T lymphocytes from the human fetus, were injected into the mouse tail vein. These lymphocytes enter the thymus tissue under the kidney and mature to T lymphocytes. The so produced T lymphocytes enter the circulation and in the lymph node present under the second and the lymph node present under the second kidney. They multiply to form a full-fledged functional immune system. It takes about 2 weeks after the transplant for the mice to display the human immune system. That means characteristics of both T lymphocytes and beta lymphocytes will be found. The human mouse Being a close animal model for the human immune system is a boon for immunologists, particularly working on AIDS. This is because the various immunological aspects of AIDS, including the possible development of an AIDS vaccine, can be explored by using human mouse. This is no good animal model for AIDS research except chimpanzee. Now let's know about the Alzheimer's mouse. The Alzheimer's disease affects about 1% of the population between 60 to 65 years old and about 30% of the population over 80 years old. This disease is characterized by progressive loss of memory and personality changes that decline in thinking, judgment, etc. The postmortem analysis of brains of Alzheimer's patients revealed plaque of dead nerve cells entangled in a protein called amyloid. This protein was purified and its amino sequence is determined. Later, amyloid precursor protein, in short APP, and its gene sequence were also identified. Alzheimer's disease is more prevalent in some families, clearly indicating a genetic basis for this disease. Transgenic mice were developed by introducing amyloid precursor gene into fertilized egg cells of mice. The synthesis of human amyloid protein and its accumulation as typical plaque in the mice brain were observed. The Alzheimer's mouse is very useful in understanding the pathological basis of the disease. Certain mutations in APP gene and the involvement of some other gene are believed to be responsible for Alzheimer's disease. Now let's know about the onco mouse. The animal model for cancer is the onco mouse. That means onco refers to cancer. First development for breast cancer, the onco mouse is useful for understanding the cancer and evolving modalities for cancer therapy. The oncogene CMYC in association with the mouse mammary tumor, in short MMT virus, was found to be responsible for breast cancer in animals. Transgenic mice were produced by introducing chimeric DNA consisting of CMYC gene and sections MMT virus into fertilized mouse egg cells. Breast cancers developed in adult female mice and trait was passed on to the offspring. Onco mouse, the mouse that is genetically altered and is susceptible for cancer, was patented by US Patent Office in 1988. In fact, it was the first animal to be patented. Now let's know about the prostate mouse. The prostate gland surrounding the urethra of male is responsible for secreting semen fluid. In the old man, particularly above 60 years of age, prostate gland gets enlarged and may become cancerous. The oncogene for prostate cancer may be identified, that is INT2. A chimeric DNA by joining INT2 with a viral promoter was prepared and introduced into fertilized mouse eggs. In the transgenic mice, so developed enlargement of prostate gland was observed. Their prostate mice were also patented in 1991. Now let's know about the knockout mouse. The basic principles underlying gene knockout have been we will have been we will discuss described. Several knockout mice have been developed. It is not an exaggeration that the knockout mouse have become as common as a test tube in the laboratory for a biotechnologist. It must be noted that not all the knockout mice are immediately useful for human health and welfare. Now let's know about SCID mouse. In that means severe combined immunodeficiency, mm, in, that is SCID, in short SCID. Again repeating, severe combined immunodeficiency is a condition with a total lack of immune system. SCID mice were developed by eliminating a single gene, and the resultant mice lost the ability to produce beta lymphocytes and T lymphocytes. The human mouse was later developed from the SCID mouse. Now let's know about knockout mouse for allergy. The receptor site on certain body cells for IgE bodies are believed to be responsible for the triggering allergy reactions. Knockout mice were developed for allergy by removing the gene encoding for receptor protein. The result is that antibodies cannot bind to cells due to lack of receptors, and the mice are unaffected by allergic reactions. It is expected that some breakthrough may occur in the near future to benefit the millions of sufferers of allergic reactions spread throughout the world. Now let's know about knockout mouse for transplantation. By using a suicide gene, the liver cells were destroyed in a mouse that already lacks immune system. Sample, sample human liver cells were transplanted in this mouse. Encouraged by lack of immune system, the human liver cells can develop. In this fashion, the organ replacement in animals is possible. Now let's know about knockout mouse with memory loss. The memory process in brain are believed to be carried out by a specialized area called hippocampus. By gene knockout technique, researchers have developed mice that lack hippocampus. These knockout mice lacked the ability to remember. Now let's know about knockout mouse with retinitis retinitis pigmentosa. By inactivating the mouse rhodopsin gene, rod cells of the retinal deteriorate in the transgenic mice. This retinal degeneration is comparable to human retinitis pigmentosa. The rhodopsin knockout mouse is useful for understanding the retinal degeneration besides evolving therapeutic strategies. Now let's know about transgenic mice for human diseases. Transgenic mice are important for the development of therapeutic drugs and possible gene therapies besides the understanding of the human disease. Many transgenic mice with human diseases equivalents have been developed and we can see. Now let's know about transgenesis in large animals. In general, transgenesis in large animals is more difficult than with mice. There are several factors for the lower efficiency of transgenesis in large animals. These include less number of eggs they and technical difficulties in handling. Besides long gestation periods to get the offspring, that means it takes about 2 years to produce a calf from a fertilized egg. Some of the early experiments to produce transgenic large animals were far from satisfactory. For instance, transgenic sheep overproducing growth hormone grow leaner with increased feed efficiency, but they are more susceptible to in fact become infertile and tend to die at the young age. All this might be due to ineffective control of gene regulation. Several improvements have been developed to produce transgenic animals with desirable characteristics. Now, biotechnologists are particularly interested to improve the quality of animals with improved resistance to diseases besides enhancing their ability to produce foods. Building a better animal being the motto. Further, production of commercial and pharmaceutical compounds by transgenic animals is also gaining importance in recent years. The protocol adapted for producing other transgenic animals is comparable with that already we discussed for transgenic mice with certain modifications. Now let's know about transgenic cattle. The mammary gland of the dairy cattle is an ideal bioreactor for producing several new proteins of pharmaceutical importance besides improving the quality and quantity of the existing ones. For instance, transgenic cow with an overexpressed casein transgene can give milk with higher content of casein. If lactase transgene is introduced and expressed in the mammary gland, milk free from lactose will be secreted. Such a milk will be a boon for lactose intolerant people who experiment indigestion and other complications after consuming normal milk and milk products. Some success has been achieved in creating transgenic cattle with improved resistance to viral, bacterial, and parasitic diseases. However, this is not an easy job due to the complexity of genetic control to combat the disease producing organisms. Attempts have been made in recent years to produce cattle with inherited immunological protection by transgenesis. Introduction of genes that encode for heavy and light chains of monoclonal antibodies has met with some success in this direction. In vivo immunization of an animal, although not yet fully successful, is ideal for disease protection. In vivo, in vivo immunization primarily involves the insertion of transgene for an antibody that specifically bind to an antigen. Now let's know about transgene sheep and goat. Transgenic sheep and goats. Transgenesis experiments in sheep and goats mostly involve the development of mammary glands as bioreactors for the production of proteins for pharmaceutical use. This is possible despite the fact that quantity of milk produced by sheep and goats is less than that of dairy cattle, that means cow or buffalo. Some proteins produced by sheep and goats have good pharmaceutical use. Now let's know about transgenic sheep with increased wool production. Keratin is the wool protein with highly cross-linked disulfide bridge. For good production of quality wool, the amino acid cysteine or its precursor methionine is required in large quantities. However, cysteine supply to sheep is always inadequate since the microbes harboring the rumen utilize it and release in the form of sulfide. This problem can be overcome by producing transgenic sheep containing bacterial genes for the synthesis of cysteine. The two enzymes synthesized by the transgenes are capable of trapping the hydrogen sulfide liberated in the intestine to produce cysteine. Thus, good supply of cysteine to the sheep improves the quality and quantity of wool. Now let's know about transgenic pigs. Transgenic pigs that can produce human immunoglobulin have been successfully developed. This human hemoglobin can be separated from pig hemoglobin by simply analytical techniques. Hemoglobin, the oxygen carrying protein of RBC, can be used as a substitute in blood transfusion experiments. In fact, hemoglobin can be stored for long period, a few months, than whole blood weeks only. Further, there is no problem of contamination like HIV, as is the case with whole blood. However, the free hemoglobin, that is naked hemoglobin, cannot transport oxygen as effectively as the hemoglobin of RBC. In addition, naked hemoglobin is easily degraded and the breakdown products cause damage to kidney. There is also exists a risk of contamination by pig viruses and other compounds to cause allergic reactions. With these limitations, the initial enthusiasm for substituting blood transfusion with free hemoglobin has remained short-lived. It is now advised not to use naked hemoglobin for transfusion when there is a heavy blood loss. However, it can be used during major surgeries for supplementing the whole blood transfusion. Now, let's know about pig in organ farms. The human organs such as heart, liver, pancreas, kidney, and lungs are in great demand for transplantation surgery. The shortage of these transplantable organs can be overcome by developing them in animals. Pig is a favorite animal for harvesting human organs. This is because the physiology of pigs is close to that of humans. Further, pigs do not carry any major infectious diseases transmissible to humans. The use of pigs in organ farming is still at the experimental stages. In the preliminary experiments, organ transplantation from transgenic pigs into primates show some promising results. The day may not be so very far for utilizing transgenic pigs as donors of human organs. Now let's know about transgenic chickens. The production of transgenic chickens is rather complicated. This is mainly because during fertilization in chickens, several sperms enter the ovum instead of one. This is in contrast to mammals where usually only one sperm enters the egg. The identification of male pronuclei that will fuse with female pronuclei is quite difficult. Further, embryonic stem cells have not been identified in chicken. Despite all these limitations, transgenic chickens have been developed. The blastoderm cells, that is from an egg, can be removed from a donor chicken. They are transplanted with transgenes, usually by liposuction with liposomes. The so modified blastoderm cells are reintroduced into the subgerminal space of irradiated blastoderm of freshly laid eggs. Some of the resulting chickens may carry the transgene. Transgenic lines of chickens can be established. Transgenesis in chicken can be used to develop low fat and cholesterol and high protein containing eggs. Transgenic chickens that are resistant to viral and bacterial diseases have also been developed. So, some attempts have also been made to develop pharmaceutical proteins in eggs of transgenic chickens. Now, let's know about transgenic fish. Several transgenic fish like catfish, salmon, trout, etc. have been developed with increase in their growth and size. There was carried out by introducing growth hormone transgene by microinjection or electroporation. The fertilized eggs with inserted transgene are incubated in temperature regulated holding tanks. The efficiency of fish transgenesis is as high as 70%. It was found that the transgenic salmon fish with growth hormone transgene were 10 times heavier than the normal ones at the end of 1 year. Now let's know about position effects. Position effect is the phenomenon of different levels of gene expression that are observed after insertion of a new gene at different positions in the eukaryotic genome. This is commonly observed in the transgenic animal as well as plants. These transgenic animals show variable levels and patterns of transgene expression. In a majority of cases, position effects are dependent on the site of transgene integration. In general, the defective expression is due to the insertion of transgene into a region of highly packaged chromatin. The transgene will be more active if inserted into an area of open chromatin. The positional effects are overcome by a group of DNA sequences called insulators. The sequences referred to as specialized chromatin structure, in short SCS, are known to perform the functions of insulators. It has been demonstrated that the expression of the gene is appropriate if the transgene is flanked by insulators. Now let's know about animal bioreactors. Transgenesis is wonderfully utilized for production proteins of pharmaceutical and medical use. In fact, any protein synthesized in the human body can be made in the transgenic animals, provided that the genes are correctly programmed. The advantage with transgenic animals is to produce scarce human protein in huge quantities. Thus, the animals serving as factories for production of biologically important products are referred to as animal bioreactors or sometimes farm animals. Frankly speaking, transgenic animals are as bioreactors can be commercially exploited for the benefit of mankind. Once developed, animal bioreactors are cost effective for the production of large quantities of human proteins. Routine breeding and healthful living conditions are enough to maintain transgenic animals. The importance of transgenic animals has already we discussed. Okay, so now we will discuss about Dolly, the transgenic clone. Dolly, the first ever mammal clone, was developed by Wilmot and Campbell in 1997. It is a sheep, that is female lamb, with a mother and no father. The, the technique primarily involves nuclear transfer and the phenomenon of totipotency. The character of a cell develop to different cells, tissues, organs, and finally an organism is referred to as totipotency or pluripotency. Totipotency is the basic character of embryonic cells. As the embryo develops, the cells specialize to finally give the whole organism. As such, the cells of an adult lack totipotency. Totipotency was introduced into the adult cells for developing Dolly. The mammary gland cell from a donor we were isolated. They were subjected to total nutrient deprivation or 5 days. By this process, the mammary cell abandon their normal growth cycle and enter a dormant stage and regain totipotency character. An ovum, that is egg cell, was taken from another Eve and its nucleus was removed to form a enucleated ovum. As the dormant mammary gland cell and the enucleated ovum were fused by Paul's electricity. The mammary cell out membrane was broken, allowing the ovum to envelop the nucleus. The fused cell, as it had gained totipotency, can multiply and develop into an embryo. This embryo was then implanted into another UV, which served as a surrogate or foster mother. 5 months later, Dolly was born. As reported by Wilmot and Campbell, they fused 277 ovum cells, achieved 13 pregnancies, and of these, only one pregnancy resulted in live birth of offspring Dolly. Now, let's know about cloning of pet animals. Some of the companies involved in transgenic experiments have started cloning pet animals like cats and dogs. Little Nicky was the first pet cat that was cloned at a cost of $50,000 by an American company in December 2004, and more cloned cats and dogs will be made available to interested parties in due course who can afford. Some people who own pet animals are interested to continue the same pet, which is possible through cloning. There is some opposition to this approach as the cloned animals are less healthy and have shorter lifespan besides the high cost factor. Now let's know about transgenic animals produced by cloning fetal cells. Fetal cells such as fibroblasts are totipotent. Fetal cell cloning was successfully carried out by some workers to produce transgenic sheep called pulli, transgenic bull calf that is gene, and other animals. Transgenes foreign genes can be inserted into the fetal cell genome to produce desired products by transgenic animals. The fibroblasts were collected from a 55-day old bovine fetus, and these fibroblasts with totipotent character are cultivated in a nutritious medium. The desired foreign genes can be introduced into fibroblast. The nucleus with the genetically altered DNA fibroblast nucleus is removed from the cell. It is then inserted into a bovine enucleated ovum. This ovum cell multiplies to form embryos. They are implanted in a surrogate mother cow to give birth to transgenic calves. The calves represent clones since they have originated from a single cell. They are transgenic as they carry the transgenes. The production of transgenic calves is a good example of nuclear transfer technology. Introduction of foreign genes can be effectively carried out in fetal cells, and so produced transgenic animals can serve as good animal bioreactors to synthesize several products of pharmaceutical importance. The progress in this direction has been rather slow due to ethical and moral issues. Now let's know about transgenic animals in xenotransplantation. Organ transplantation, that means kidney, liver, heart, etc. in humans, has now become one of the advanced surgical practices to replace the defective, non-functional or severely damaged organs. The major limitations of transplantation is the shortage of organ donors. This often results in long waiting times and many unnecessary deaths of organ failure patients. Xenotransplantation refers to the replacement of failed human organs by the functional animal organs. The major limitation of xenotransplantation is the phenomenon of hyperacute organ rejection due to host animal system. The organ rejections is mainly due to some reasons. Two causes are there. So first cause is the antibodies raised against the foreign organ. Second is activation of host complement system. Now let's know about pigs in xenotransplantation. Some workers are actively conducting research to utilize organs of pigs in xenotransplantation. It is now identified that the major reason for rejection of pig organs by primate is due to the presence of a special group of disaccharides in pigs and not in primates. The enzyme responsible for the synthesis of specific disaccharides in pigs has been identified. It is alpha 1,3 galactosyl transferase present in the pigs and not in the primates. Scientists are optimistic that knockout pigs lacking the gene encoding the enzyme alpha 1,3 galactosyl transferase can be developed in the next few years. Another approach is to introduce gene in primates that can degrade or modify gal alpha 1,3 gal disaccharide groups, that is of pigs. This will reduce immunogenicity. Besides these topics, there are other strategies to avoid hyperactive organ rejection by the host in xenotransplantation. Expression of antibodies against the pig disaccharides. Expression of complement, that means inactivating protein on the cell surface. By this approach, these two approaches, it may be possible to overcome immediate hyperactive rejection of the organs. The next problem is the delayed rejection, which involves the macrophages and natural killer cells of the host. Another concern of xenotransplantation is that the endogenous pig viruses could get activated after organ transplantation. This may lead to new genetic changes with unknown consequences. The use of transgenic animals in xenotransplantation is only at the laboratory experimental stages involving animals. It is doubtful whether this will become a reality in the near future. There is a vigorous debate concerning the ethics of xenotransplantation, and the majority of general public are against it. Now let's know about transgenic organisms to interrupt disease cycles. Several human diseases have a second host in the life cycle of the parasite. Transgenesis will be useful for disease control by interrupting the life cycle. Attempts are being made in the recent years in this direction. Although the success has been limited, and transgenic organisms may soon serve as environmental replacements to control several diseases. Now let's know about transgenic snails. Schistosomiasis is a parasitic disease caused by schistosoma, which develops in the snails. The parasite infects humans by penetrating their skin when contacted in water. It is estimated that about 100 million people worldwide are the victims of schistosomiasis. This disease is characterized by chills, fever, intestinal ulcerations, and diarrhea. By developing transgenic snails, it is possible to interrupt the life cycle of schistosoma. Some workers have attempted to create transgenic snails that will resist the invasion of the parasite. If such snails are released into the environment, they will break the life cycle of schistosoma. Now let's know about transgenic mosquitoes. Mosquitoes are responsible for the transmission of malaria. Some workers have identified certain critical genes in mosquitoes, that is Anopheles genes, that transmit Plasmodium species malarial parasite, that are responsible for harbor and transmission of the parasite. Theoretically, it is possible to alter these genes and produce transgenic mosquito. If such mosquitoes are released into the environment in large numbers, they will dilute the population of native mosquitoes and halt the transmission of malarial parasite. Another approach to control malaria is to understand vector incompetence, that is the genetic basis of the vector to sustain a parasite, and how to alter it so as to make the vector inhospitable for the parasite. Some success has been achieved in the developing transgenic mosquitoes with vector incompetence. If such mosquitoes are released into the environment, the life cycle of the malarial parasite will be interrupted. Now let's know about transgenic tisets flies. A protozoal disease, African sleeping sickness, is transmitted by tsetse flies. This disease affects the nervous system and is often accompanied by coma. Transgenic T-sets flies were developed with a novel approach. Researchers found a protein that can kill disease-producing protozoa. They identified the protein encoding gene and inserted it into the bacterial of T-sets fly gut. These bacteria produce anti-protozoal protein that kills T-setse flies. In this manner, the disease transmission can be prevented. Now, let's know about transgenic bollworms. Bollworms are the caterpillars that damage the cotton crop. Researchers have developed transgenic bollworms with a suicide gene. When released into the environment, the transgenic bollworms will mate with wild bollworms and produce caterpillars that die. The cotton crop can be saved from the damage. Now let's know about transgenic mayflies. Mayflies, that is Mediterranean fruit flies, destroy fruit and coffee crops throughout the world. Attempts are being made to develop transgenic mayflies to replace wild mayflies and save the crops. Finally finished, I have finished the transgenic animal portion. Any questions regarding this, you can ask me in the comment box, and subscribe my channel to know more topics about biotechnology. And here, if anyone searching is studying BSc Biotechnology, MSc Biotechnology, or preparing for CSIR UGC NET, can refer my channel, and I will definitely give every day, Monday to Friday, every day giving biotechnological topic. One topic I'll upload. So you can follow my channel, and to know more updates, you can subscribe my channel. Thank you.